when the news of this, supposed new illness and later announced as a pandemic came out, I wanted to say, okay, well, let me go look at the paper that identified this virus so I can see what it is and, how they did it. And what I realized is that the methods that they used in their experiments actually couldn't even show a virus. And it was really strange because in the title of their papers, they used the of a novel coronavirus, right? A new, it's never been seen or identified before or known to exist. And then, but they don't actually use the common human meaning of the word isolation, right? Which is to separate from other things. That has never been done with this virus or other viruses. Instead, they basically add a sick person's secretions, right? In this case, from a BAL fluid sample, which is just fluid from the lungs.
Right.
They put it into a foreign tissue culture of usually monkey kidney cells, and then they add a few poisons. And interestingly, they choose like antibiotics, which they justify by saying that, it would kill any bacteria that would contaminate the sample, but they choose antibiotics that are nephrotoxins, and then they're using kidney cells in the culture. So like amphotericin and gentamicin are common ones that they use. Then they add foreign proteins in the form of fetal calf serum to the cell culture, and then they use minimal growth media, which essentially starves the tissue. And then they observe that that tissue decays. And when tissue decays, the cells break apart into particles. That's what we see under the microscope time and time again. And that's all they're seeing. When they show a picture that they say is this virus, there's no way they know what they're looking at. These are just cells disintegrating. And they're throwing out all kinds of particles. And so it's a complete misinterpretation. And they also, by the way, they don't do a control experiment where they mix everything the same, except they have the lung washings from a healthy person.
Right.
And, if they did that, I'm sure what you'd see is the same exact destruction or decay of the tissue in the tissue culture. And then you'd not be able to differentiate, and you say the experiment is negative. And then you go on and devise another experiment. And by the way, they have the experimental methods to actually isolate viral particles out of that lung fluid. And they've done it for exosomes, which are particles that come from our own cells when they're damaged, especially. So they can purify those. They've purified bacteriophages, which are particles that exist in pure bacteria cultures under certain laboratory conditions, but they're the same size as viruses, and they are sometimes thought to be viruses of bacteria. And they can purify those, no problem, and there are dozens of papers where they've done that. So what we have here really is a failure of science to see the results when, and we really know this is already the case because you have like Professor Ioannidis from Stanford, who's one of the world's leading epidemiologists, and he published a paper back in 2015 explaining why more than half of all published research findings are false. So why is it so hard to fathom? That's one of the most cited papers in the literature. So to fathom that these virus papers are in that more than half that are false has a high probability, right? That probability is greater than 50% for any
Virtually
paper.
guaranteed.
So it's not really that much of a stretch. And then what they do is they kind of fake satisfying Koch's postulates by taking a filtrate of the toxic cell culture, put it in experimental animals, and these are like sometimes even transgenic animals, so they're not even normal animals, but then they can't produce the same illness. The animals don't even get sick, really. They don't have the same symptoms or autopsy findings or anything. But yet the paper says that they proved this, but they didn't. And then you have the genetic sequences, where this is the biggest quagmire in terms of difficulty to understand, because just understanding the basics of what DNA is, right, is very challenging for most people. And there aren't a lot of good materials out there explaining it at that level, that you're not a college chemistry major, And so when you look at this, it's really hard to figure out, but there are some clues. So one thing is they have like hundreds of different variants of the genetics that they say is this virus, right? So if there's so much variability, how could it actually be a real thing? Because there's not this much variability in known organisms. But take that aside, let's just look at how they found this so-called sequence. Well, they didn't take a viral particle and pull out its genetic material. And that would be really the only way to know what you've got. And that's how they, sequenced the human genome. They took DNA from human cells, right? Very straightforward. But in this case, they actually take the same lung fluid or snot, which, has hundreds of different kinds of cells in there, different species of bacteria, fungi, different kinds of human cells, right? Free genetic material that has been found in sputum. And then, whatever else might be a contaminant. So it's taking this messy sample, and they design these little tags that look for short pieces of genetic material after they kind of release all the genetic material and remove the cellular debris. And these little primers and probes that are the tags, they actually are found in hundreds of different bacteria species and different human chromosomes. So they're not like unique to anything. And they've never been shown to come from this virus because they've never actually had the virus in a pure form where they could pull it out. So what they do is they use these little tags, and then they extend those sequences to whatever they find, and then they sequence those things, and they have no idea what they are, where they come from, but they put all that data into a computer. And the computer mixes it up in a model that is one long string. And there are gaps, of course, and the gaps are just filled in by the computer according to the instructions that are programmed in. So they might come from some database of other unrelated viruses, or they say they're related, but how do they know? Because they never saw what they look like or the particles, or did the analysis taking it from the organism. So this is sometimes referred to as an in silico genome, and it's purely theoretical from an organism that is also theoretical. And they keep building upon this, right? The assumption that they had a real thing that they showed, and that they demonstrated that that thing causes a new illness. And everything else is predicated. So they say, oh, here's the genome of the thing. Except they don't tell you, well, we didn't actually get it from the thing. We got it from 20,000 little pieces that we put together in a computer program.
Wow.
Right?
Wow.
That's actually what they really did. And that's why there are hundreds of different sequences, because each time they do it, they're just getting random stuff. And when they put random stuff together, and they try to put it together in the same way, but it's still different. So they say that these are just mutations or variants, but really it's all junk.